Welcome friends. In the previous lecture we had seen about the various factors as well as various formulation components to formulate pharmaceutically stable aerosols and the various applications for which they can be used. In the current lecture we will be looking into various evaluations which can be used to predict the stability of the aerosols which are used for both pharmaceutical as well as cosmetic applications. So, in the current lecture we are going to understand the basic components and mechanisms of the pharmaceutical aerosol systems. We can be looking into the significance of evaluating aerosol dosage forms quality, safety and efficacy. Also we will be identifying various tests such as leak testing, spray pattern and dose uniformity. We will also understand the methods to determine particle size and plume geometry. Also the importance of valve and container integrity from the aerosol performance point of view will be looked, and also the standard conditions and protocols for stability testing of the aerosols will be discussed. So, the quality control of pharmaceutical aerosols involves looking into the quality aspects of all the components starting from propellants to valve, actuator and dip tubes. So, a sample is removed and sent to laboratory where its vapor pressure is determined and compared to specifications for the propellants. The gas chromatography is used to determine the identity of propellant, and when a blend of propellants is used, it is used to determine its composition. The purity and acceptability of the propellants is tested by moisture, halogen and non-volatile residue determinations. Monographs for various propellants are written and continuously being upgraded in the various pharmacopoeias. The valves, actuators and dip tubes are also subjected to physical and chemical inspection. The examination at this point must determine whether the valves are fit to be used. They are sampled according to the standard procedure as found in military standards. So, the different solutions for various propellants are mentioned in the table 1. Then the test which is used to provide means for determining the acceptance of metered dose aerosol, the magnitude of valve delivery and the degree of uniformity between the individual valves as related to acceptance of any given lot of metered aerosol valves is compared. So, the testing procedure involves selecting 25 valves per shipment using standard sampling methods. The valves are attached to containers filled with specified test solution. A button type actuator with the orifice greater than or equal to 0.021 inch is used and fixed. The containers are placed at 25° until temperature equilibrium is reached. Then each valve is actuated fully for more than 2 seconds or equal to 2 seconds after complete spray. The test that is the actuation is repeated for 10 times to prime the valve. Each unit is weighed to its nearest milligram. 2 separate full actuations of more than 2 seconds are performed and weighed after each actuation. The delivery is calculated by weight difference for each actuation. Then each delivery weight is divided by the test solution specific gravity to get microliters per actuation. So, this is the flow chart of the testing procedure for the quality control of propellants. The valve acceptance is also important. The test procedure applies to 2 categories of metered aerosol valves having the following limits. For valves delivering 54 microliter or less, the limits are within plus minus 15%, and 55 to 200 microliter, the limits are between plus minus 10%. So, of the 50 individual deliveries, if 4 or more are outside the limits for the specified valve delivery, the valves are rejected. If 3 individual deliveries are outside the limits, another 25 valves are sampled and the test is repeated. The lot is rejected if more than 1 delivery is outside the specifications. Further, if 2 deliveries from one valve are beyond limits, another 25 should be taken. The lot is accepted if not more than one delivery is outside the specifications. Containers are also sampled according to the standard sampling procedure. Both uncoated and coated metal containers must be examined for defects in the lining. Several quality control aspects include specifications for the degree of conductivity of an electric current as a measure of the exposed metal. Glass containers must be examined for flaws. Dimensions of the neck and other parts must be checked to determine conformity to the specifications. Also, the weight of the bottle must be determined. The weight checking is usually accomplished by periodically adding to the filling line tied empty aerosol containers, which after being filled with concentrate are removed and then accurately weighed. The same procedure is used to check the weight of the propellant that is being added. When a propellant blend is being utilized, checks must be made to ensure a proper blend of the propellants. As a further check, the finished container is weighed to check the accuracy of the filling operation. Leak testing, a means of checking the crimping of the valve. It must be available to prevent defective containers due to leakage. For metal containers, this is accomplished by measuring the crimp dimensions and ensuring that they meet the specifications. Final testing of the efficiency of the valve closure is accomplished by passing the filled containers through the water bath. Periodic checks are made of the temperature of the water bath and the results are recorded. The spray testing is done to clear the dip tube of the pure propellant, to clear the dip tube of the pure concentrate for products filled by pressure under the cap or around the stem, and to check the defects in the valve and the spray pattern. For metered valves, it serves to prime the valve so that it is ready for use by the consumer. The aerosols are pressurized packages and many tests are necessary to ensure proper performance of the package and safety during use and storage. These regulations impose limitations on the pressure within the container, flash points, flame extinction and flammability. The provisions of the Hazardous Substances Labelling Act and the Food, Drug and Cosmetic Act must be applied. In addition to the federal regulations, many local officials impose further restrictions upon aerosols. So, the evaluation of aerosols can be done based on flammability, physicochemical properties, performance and the biological characteristics. The flammability and combustibility. The flash point is determined by the use of tag open cup apparatus. The aerosol product is chilled to a temperature of 25° Fahrenheit and transferred to the test apparatus. The test liquid is allowed to increase slowly in temperature and the temperature at which the vapors ignite is taken as the flash point. Flame projection. This test indicates the effect of an aerosol formulation on the extension of a opened flame. The product is sprayed for about 4 seconds into a flame. So, when you spray the aerosol on the flame, to what length the flame gets extended is measured. The physicochemical characteristics, the vapor pressure is measured. The pressure can be measured simply with a pressure gauge or elaborately through use of a water bath, test gauges and special equipments. This is important to measure so that the pressure variation from container to container can be determined, since excessive variation indicates presence of air in the headspace. A can puncturing device is available for accurately measuring vapor pressure. Density is determined through the use of a hydrometer or a pycnometer. These methods, which have been used for the density of non-aerosols, have been modified to accommodate liquefied gas preparations. A pressure tube is fitted with metal flange and a hook valve, which allows for the induction of liquids under high pressure. The hydrometer is placed into the glass pressure tube. Sufficient sample is introduced through the valve to cause the hydrometer to rise halfway up the length of the tube. The density can be then read. The specific gravity can be determined through the use of high pressure cylinder of about 500 milliliter capacity. The moisture content can be measured by the Kalb-Fisher method and gas chromatography. The propellants can also be identified by the gas chromatography and infrared spectrophotometry. The aerosol valve discharge rates can be performed, and this is determined by taking an aerosol product of known weight and discharging the contents for a given period of time using standard apparatus. By reweighing the container after the time limit has expired, the change in weight per time dispensed is the discharge rate, which can be expressed as grams per second. The spray patterns are also measured, and a method for comparing spray patterns obtained from different batches of material or through the use of different valves is taken into consideration. The method is based on impingement of the spray on a piece of paper that has been treated with a dye talc mixture. Depending upon the nature of aerosol, an oil soluble or water soluble dye can be used. The particles that strike the paper cause the dye to go into solution and to be absorbed onto the paper. This gives a record of the spray, which can then be used for comparison purposes. To control the amount of material coming into contact with the paper is attached to a rotating disc that has an adjustable slit. The dosage delivered with meter valves can also be measured and should also be measured. So, several points must be considered. First is the reproducibility of dosage each time the valve is depressed, amount of medication actually received by the patient. The reproducibility of dosage may be determined by assay techniques, whereby 1 or 2 doses are dispensed into a solvent or onto a material that absorbs the active ingredients. Another method that can be used involves accurate weighing of filled container followed by dispensing of several doses. The container can then be reweighed, and difference in weight divided by the number of doses dispensed can be used as a measure to find the average dose. The net contents can be measured using several methods to determine whether sufficient product has been placed into each container. The tared cans that have been filled onto the filling line are reweighed, and the difference in weight is equal to the net contents. The other method is a destructive method and consists of weighing of full container and then dispensing the contents. The contents are then weighed with provision being made for the amount retained in the container. Other modifications consist of opening the container and removing as much as the product possible. The National Bureau of Standards has issued a method that can be used for foam type, low viscosity, high viscosity and food aerosols. These methods standardize the manner in which the containers are to be dispensed. Foam stability. The life of a foam can range from a few seconds for some quick breaking foams to 1 hour or more, depending upon the formulation. Some methods include a visual evaluation, time for a given mass to penetrate the foam, time for a given rod that is inserted into the foam to fall, and the use of the rotational viscometers. Particle size determination, and the cascade impactor and light scatter decay methods are widely used for the particle size determination of aerosols. The cascade impactor operates on the principle that in a stream of particles projected through a series of nozzles and glass slides at high viscosity, the larger particles become impacted first on the lower velocity stages, and the smaller particles pass on and are collected at higher velocity stages. The figure illustrates a unit suitable for analysis of particles whose diameters range from 0.1 to 30 microns. The particle size of epinephrine aerosols was determined using the light scatter decay method. In this technique, as the aerosol settles under turbulent conditions, the reduction in the light intensity of a Tyndall beam passing through the aerosol is measured. The mean median diameter of the aerosol was found to be between 2.7 to 3.5 microns. The biological testing of the pharmaceutical aerosols is essential to evaluate their therapeutic efficacy, safety and site-specific drug delivery. The purpose of biological testing is to determine the therapeutic efficacy in the target organs like lungs, nasal mucosa, to ensure the safety of aerosolized drugs and excipients, evaluate the drug bioavailability and pharmacokinetics via inhalation route, also to establish dose-response relationships for optimal clinical outcomes. These tests ensure that the aerosol formulation delivers the intended pharmacological effect upon administration. The types of biological testing include both in vivo and in vitro. These in the in vivo biological testing, these are laboratory based tests conducted on cultures, tissue models or simulated biological environments. Cell viability and cytotoxicity studies using lung epithelial cell lines can be done. Permeation studies across lung tissue models can also be done. The studies can also be performed on animal models to study the systemic as well as local effects of the aerosolized drug. Pulmonary deposition and distribution studies using radio-labeled aerosols can be done. Bronchodilation testing in asthma model, example guinea pigs or mice sensitized with ovalbumin can be done. Histopathological examination for lungs and other organs for toxicity is done. Pharmacokinetic studies which involves ADME is also important while doing the biological testing. Pharmacodynamic assessment to determine the therapeutic outcomes like reduction in airway resistance or inflammation is also important part of the biological testing. The factors which can influence the biological testing and its outcomes are the particle size and distribution. The optimal range pulmonary deposition is 1 to 5 micrometer. Larger particles greater than 5 micrometer may deposit in the oropharynx, reducing efficacy. Very fine particles less than 1 micrometer may be exhaled, leading to loss of the dose. Influences the site of deposition, that is upper or lower airways, and hence the therapeutic effect which is possible because of the variation in the size of the particles. The formulation stability is another factor. Physical and chemical stability of the drug during storage and aerosolization is crucial. Aggregation or crystallization of particles can alter the aerosol performance. Use of appropriate excipients like surfactant stabilizers can enhance the shelf life and the biological efficacy. Further, the type of device used can also have an impact. Metered dose inhalers, dry powder inhalers or nebulizers deliver aerosols differently. Each device has specific performance parameter like plume geometry, spray velocity, inspiratory flow requirements. A mismatch between the device design and patient capability affects the drug deposition. The dry powder inhalers depend on patient's inspiratory effect. Nebulizers are preferred for hospital or pediatrics due to easy inhalation. Device design must thus match patient population and the drug characteristics. The patient inhalation technique, that is incorrect use, leads to poor drug deposition and lower efficacy. Lack of coordination in MDIs can result in oropharyngeal deposition. Inadequate inspiratory volume in DPIs can lead to ineffective dosing. Training and education are thus important for proper inhaler use. Variability in technique can skew biological testing results. Spacers or holding chambers can help improve delivery. Lung physiology and disease state can also have an impact on the biological testing outcomes. Mucus hypersecretion, inflammation or bronchoconstriction can block airflow. Disease alters mucociliary clearance, affecting residence time. Lung pH, enzyme levels and surfactant may influence drug absorption. Asthmatics and COPD patients have different deposition due to limited proximal airways. Lung function like tidal volume affects the aerosol penetration. Biological testing must consider thus the disease models and the patient variability. The therapeutic activity of the aerosols are widely used for delivering the drugs directly to the respiratory tract, offering rapid onset of action and targeted therapy with minimal systemic side effect. The therapeutic activity of aerosol depends on the site of deposition, that is, the particles must reach the desired region of lungs for optimal effect. Drug absorption. Aerosolized drugs can be absorbed systemically or can act locally on the airway tissues. Dose uniformity. Consistent dosing ensures reproducible therapeutic outcomes. Onset and duration of action. Aerosols provide fast relief in conditions like asthma, with effects ranging from short to long active depending on formulation. Local versus systemic effects. While many aerosols act locally like bronchodilators, and some are designed for the systemic delivery like the insulin and or the pain relief preparations. Toxicity testing should include both topical and inhalation effect toxicity. The aerosols applied topically may be irritating to the affected area and may cause chilling effect. The degree of chilling effect depends on the type and amount of propellant used. The inhalation toxicity must be considered even though the product may be intended for topical administration. This can be accomplished by exposing test animals to vapor sprayed from an aerosol container. Stability testing is also done. The testing of the aerosols must cover 3 areas for stability testing, that is concentrate and propellant, container and valve. The evidence of decomposition or deterioration in any of these areas could result in the ineffective product formulation. For the concentrate and propellants, immediately after preparation, several of the important physicochemical constants of the product are determined, like vapor pressure, spray rate of valve, pH, density or specific gravity, refractive index, viscosity, total weight, assay of the active ingredients and like. The contents of the container are removed by chilling the contents to a temperature of 0 degree Fahrenheit or less and opening the container. The container is then examined for signs of corrosion. These changes can be detected without much difficulty, since attack upon tin plate, tin free steel or aluminum is generally visible to the naked eyes under a microscope. Also can be done. For these containers that have internal lacquering, the examination must ensure that the lacquer is not softened, dissolved or peeled or blistered by the concentrate. When glass is used for the container, an examination of the container can be omitted. Plastic containers may require special testing to determine whether leaching or absorption has taken place. The valve should be examined to ensure that it is functional and will satisfactorily dispense the product and be easily closed. This can be readily determined by the dispensing of the product. The valve cup should be examined for evidence of corrosion. The various valve subcomponents should also be examined for evidence of softening, cracking, elongation or distortion. Elongation and cracking of the dip tube should be noted and if present, corrected. The aerosols for pulmonary drug delivery. The 2 possible mechanisms for delivery of drug to the lungs include an aerosol or a direct instillation. The most commonly used in the aerosol, which consists of finely divided liquid droplets or solid particles in a gaseous suspension. A device which generates an aerosol is termed as an atomizer. An atomizer may be electrically, pneumatically or mechanically powered. The main types of devices used at present to produce aerosols are nebulizers, metered dose inhalers or dry powder inhalers. The nebulizers can be divided into 2 main groups, pneumatic and electric. An electric generator, that is ultrasonic generator, operates from an electric source, while a pneumatic generator, hydrodynamic and jet, derives its power from the pressurized gas source. The ultrasonic nebulizer consists of a piezoelectric crystal which produces high frequency sound waves in the liquid in the nebulizing unit. The surface waves produce small droplets which are conducted away by an upstream air for inhalation. The hydrodynamic nebulizer uses a system that prepares a film of water for aerosol formation by flowing it over a hollow sphere. A small orifice in the sphere expels gas at supersonic velocity. The high velocity gas ruptures the thin film of water and produces a continuous dispersion of fine liquid particles. A gas cylinder or compressor supplies the gas pressure. In case of jet nebulizer, high velocity gas flow is directed over a tube that is immersed in a water reservoir. The expansion of the driver gas decreases the pressure over the tube, which draws the formulation into the gas stream. The high shear rate in the jet stream then nebulizes it. The properties of nebulizers may vary. While all produce droplets with sizes in the range of 1 to 10 micrometer, they may vary significantly in the droplet size distribution and pulmonary distribution. The meter dose inhalers, the drug is usually a polar solid which has been dissolved or suspended in a non-polar liquefied propellant. If the preparation is a suspension, the powder is normally micronized by fluid energy milling and the suspension is stabilized by addition of a surfactant. Lecithin, oleic acid, span, tween surfactants can be used. The meter dose inhalers are most commonly used drug delivery system for inhalation. The propellants have high vapor pressure of around 400 kilopascals at room temperature, but since the device is sealed, only a small fraction of the propellant exists as a gas. The canister consists of a metering valve crimped onto an aluminum can. Individual doses can be measured volumetrically by a metering chamber within the valve. Each MDI canister can hold between 100 to 200 doses, between 20 picogram and 5 mg of drug, which is released within the first 0.1 seconds after actuation. The aerosol consisting of propellant droplets containing drug is delivered from the actuator mouthpiece at a very high velocity, probably about 30 meters per second. There is a partial 15 to 20% evaporation of propellant prior to exit from the atomizing nozzle known as flashing, and further breakup of droplets beyond this point caused by the violent evaporation of the propellant is there. This results in a wide droplet size distribution from 1 to 5 micrometer. Only 10% of the particulates delivered in a single dose released by an MDI actually reach the lung, since the bulk of it impacts in the oropharynx and the mouthpiece. A metered propellant-free intranasal pump has been introduced to deliver flunisolide, an effective steroidal agent in the relief of symptoms associated with seasonal or perennial rhinitis. The pump permits the administration of a metered dose of steroid without using propellants, which by their cooling effects often cause smarting and irritation to the nasal mucosa. The metered aerosol pump also ensures accurate dosage and eliminates many of the administration problems associated with the nose drops. The concept of propellant-free metered delivery offers a new dimension to intranasal delivery of potent therapeutic agents. The dry powder inhalers. The environmental concerns surrounding the use of chlorofluorocarbons have led to a surge of interest in dry powder inhalers. The dry powder inhalers rely on inspiration to withdraw a drug from the inhaler to the lung, and hence the effect of inhalation flow rate through various devices has been extensively studied. It is recommended that the patients inhale as rapidly as possible from the devices in order to provide maximum force to disperse the powder. The quantity of drug and deposition pattern varies enormously depending on the device. Rotahaler used individual capsules of micronized drug which were difficult to handle. Modern devices use blister packs like discus or reservoirs like Turbuhaler. The drug delivery systems intended for deposition of medication into the nasal passages have been used as a most effective means of administering drugs intended to for both the local and systemic delivery. Until recently, the modes of administering intranasal preparations were limited to nasal drops, non-pressurized nasal sprays, mist, inhalants, intranasal gels, creams and ointments. The intranasal aerosols offer numerous advantages, including the delivery of metered dose of drug, excellent depth of penetration into the nasal passageway, reduced droplet or particle size, lower dosage than comparable systemic preparations. The major difference between the inhalation aerosol product and an intranasal aerosol product is the design of the adapter. The nasal adapter are considerably shorter and narrower, minimizing propellant vaporization before contacting the mucosa. This results in lower percentages of smaller particles, which is desirable because this decreases the number of particles entering the respiratory airways. Solution emulsion type of nasal aerosols are also regarded as vehicles for delivery of the medication through the intranasal passage. However, they are difficult to formulate. These forms of pressurized aerosol nasal preparations introduce additional problems such as vehicle viscosity, irritation from additives and leakage from the nasal passageways. So, to conclude, proper evaluation of the pharmaceutical aerosols ensures safe and effective drug delivery. Multiple parameters such as dose uniformity, spray pattern and particle size must be tested. Stability studies are crucial to assess the short shelf life and performance over time. Integrity of the container closure system directly impacts the product reliability. Overall, systematic evaluation enhances product quality and patient compliance. Thank you very much for your patient hearing. Hope you had a fruitful understanding of the pharmaceutical aerosols.